FAIM for fast apoptosis inhibitory molecule. So this speaks to the issue of apoptosis and immune destruction. So fast apoptosis inhibitory molecule is a highly conserved, uniquely structured, widely distributed gene cloned from B lymphocytes about 10 years ago. It opposes cell death, so it's anti-apoptotic, and it boosts some forms of receptor signaling, so it's signal enhancing. These functions of FAIM could contribute to the viability and/or growth activity of neoplastic tissues. And the punchline is that certain B lymphoid malignancies indeed do express elevated levels of FAIM, which may contribute to their abnormal characteristics. So a couple of questions about FAIM that I want to discuss over the next 20 minutes. The sequence of FAIM was published in 1999. How did it take us 10 years to make the connection between FAIM and malignancy? And then how does FAIM function and how is it regulated? So our work began a number of years ago when we were studying apoptosis in B lymphocytes mediated by the death receptor FAS or CD95. And we found that stimulating some receptors such as CD40 produced an increase in FAS expression and susceptibility of B cells to FAS-mediated apoptosis. However, stimulation of other receptors like the B cell antigen receptor opposed the action of FAS apoptosis and so induced a state of FAS resistance. And this is an example of what that looks like. This is an apoptosis assay conducted on primary B cells stimulated with CD40 ligand that induces sensitivity to apoptosis, or CD40 ligand and anti-immunoglobulin antibody, where the anti-Ig cross-links the B cell receptor and produces resistance to fast killing. So CD40 ligand sensitive B cells show about 40% specific cell lysis in this assay, whereas those stimulated with CD40 ligand and anti-Ig are now resistant to fast killing. So we hypothesized then that the induction of fast resistance through the B cell receptor engagement might be due to induction of a new gene. And so we carried out differential display, a technique that was common at the time to identify new genes in different populations of cells. And we compared fast-sensitive B cells with fast-resistant B cells, and indeed we did clone a new gene, a northern blot of which is shown here. So here are B cells stimulated with CD40 ligand alone. They show minimal expression of this new gene, FAIM, but after stimulation with anti-immunoglobulin antibody, FAIM expression is markedly increased. And this gene is highly evolutionarily conserved. FAIM encodes a 179 amino acid protein in both mouse and human, whose sequence is unique. It doesn't match any other known gene, and it contains no known effector motifs. However, it's highly homologous among species. It's highly evolutionarily conserved. This is mouse FAIM, human FAIM, worm FAIM, and fly FAIM. They show marked areas of identity in the amino acid sequence, but no homology with any other genes or proteins. One way to get at the possibility of effector motifs when there's no sequence homology is to look for structural homology. And so we collaborated with Gerhard Wagner at Harvard to look at the NMR structure of FAIM, and this is the result of some of that work, where he found that the C-terminal domain was well structured, and there was a separate N-terminal domain which was less well structured. The C-terminal domain is a non-interleaved beta sandwich canted at 30 degrees, which is very unusual, and the bottom line is that the structure of the C-terminal region is unique. There's no sequence homology with anything else, and there's also no structural homology between FAIM and any other protein.